Good afternoon, ladies and gentlemen. Welcome to Agrisera workshop, Tips and Tricks of Antibody Production and Western Blot, How to Obtain Good Results. My name is Joanna Porankiewicz-Aslund and I am Technical Support Manager at Agrisera, where I have been working for the last 20 years. Agrisera is located in northern Sweden and have been working with antibody production for the last 30 years. Let's take a look at antibody molecule. Just to remind you, it is a Y-shaped protein which consists of heavy and light chains, and this is what you often see when you purchase secondary antibodies, which are made to heavy and light chains. The most interesting and important for antibody recognizing an antigen is so-called antigen binding domain. And that's where the binding occurs, and it is through so-called epitope, which is varying from 3 to 15 amino acids. So an antibody is recognizing a sequence of 3 to 15 amino acids. And it can be a linear sequence, so an epitope can be linear, which is just a subsequent, which are just subsequent amino acids in protein sequence. And with such epitopes, we're working with in techniques like Western blot immunohistochemistry, where proteins are either fully or partially denatured. We also have so-called discontinuous epitopes, which are created by amino acids brought in the 3D structure of a protein. And these epitopes are recognized by antibodies in techniques where native proteins are recognized, so like immunoprecipitation, chip, and flow cytometry. There has been a study done by a Swedish group some years ago. They have been mapping epitopes, and one can ask a question: which epitopes are more common? Are they linear epitopes, so stretches of amino acids in protein sequence, or conformational epitopes, discontinued epitopes? And the group has analyzed antibodies produced by Agrisera to different cancer proteins. And afterwards, specific epitopes were mapped. And it appeared that most of these antigens have been actually stimulating production of antibodies to linear epitopes. There were just very few cases where the antigens produced more of antibodies to discontinued epitopes. We also had cases in our experience of antibody production where an antigen has been injected to 2 different animals, and one of them produced antibodies useful for Western blot, while the other one produced antibodies useful for immunoprecipitation. When we consider antibody production, we're often asked a question: which of these 3 components is most important for successful antibody production? And actually, all 3 of them are of crucial importance, so antigen, host, and testing, because all 3 of them combined together will result in successful antibody production. So let's take a look at each of them. Antigen. When we develop antibodies to proteins, we are working, of course, with a sequence. And antigen can be either a whole protein, which can be nowadays mainly recombinant, full length, or just partial, or it can also be, an antigen can be a native protein purified from tissue. Although these kind of antigens are commonly not used so frequently. A very popular way to produce the antibodies is to use synthetic peptides, which can be 7 to 30 amino acids long, and due to the fact that they are so small, they need to be conjugated to a carrier. So much larger molecule, in this case, most commonly used is KLH, He-lymphed hemocyanin from a mollusk, which has a size of over 1 million Dalton, and through the cysteine group, which is N on C or N terminal on a peptide, the peptide is attached to a carrier. The carrier is spiked with the peptide, which we use to elicit a specific immunological response. Of course, antibodies will be produced to both to the peptide and to the carrier. And when the peptide sequence is chosen by our peptide chemist, it can be located in different parts of the protein. Although one has to remember that, of course, the peptide which is chosen for immunization has to be located outside the signal peptide. Also, if there are any processing sites, they have to be avoided. Such a peptide can be either N or C terminal or derived from the inside of a protein. But location of this peptide will really influence in which techniques an antibody can be applied afterwards. If we want an antibody to work in immunoprecipitation, the peptide, which is chosen for antibody production, should be from the exposed part of the protein. So this is an example here of different peptides which can be chosen to produce antibodies to a photosystem II protein PSBA. Of course, peptides are chosen as antigens if we work with protein families with many isoforms, for example, where we want to distinguish between different isoforms. We can also choose peptides if we want to a resulting antibody to cover a wide species range. So peptides are very useful and very good for antibody production, especially for proteins of high or medium abundancy. If we work with proteins, if we want to produce antibodies to proteins of low abundancy, in such cases it's better to choose longer protein fragments, for example, up to 100 amino acids. But one has to keep in mind that such sequence has to be unique. It has to be blasted to check conservation level with other proteins from a given species under investigation. Of course, we can also use recombinant proteins, which can be either tagged or cleaved. If a recombinant protein is cleaved, in such cases, the yield is going to be, it can decrease the yield. It can be full length or partial, but still, if we use a full length protein, we need to make sure that that sequence is absolutely unique. So recombinant proteins may not be so useful to produce antibodies to detect proteins from families with many isoforms. When it comes to a buffer in which such recombinant protein is provided, it can contain actually 4 molar urea. Protein concentration, the higher the better for the animal because it will lower the volume per injection. If there are any purity issues, the protein can be separated on a gel and afterwards excised as a gel piece, homogenized and injected. This will help to remove contaminants. But we need to be sure that the band which has been injected contains only protein under investigation, or protein to which we would like to produce the antibodies. When it comes to amounts which are required for immunization, for 2 animals we will need 800 micrograms. But this is just a standard amount. Much lower amounts can be also injected. Some of these slides contain links, and if you're interested to receive this presentation afterwards, please send me an email and I will send you a PDF. Back to antigens, it can be also native protein purified from tissue. However, this format is not so commonly applied, as purifications of native proteins are quite laborious and time-consuming, and we need to be really sure that the preparation which is sent for antibody production contains only protein which we would like to detect in the Western blot later. The important point here is that proteins can be either soluble or precipitated. Actually, precipitated proteins will make pretty good antigens for antibody production. But of course, if antigen is somehow denatured, there is less chances that resulting antibodies will recognize the native protein. You need to remember that whatever is used for antibody production needs to be of a really good and high purity. So, just to remind you that if we produce antibodies to a whole protein, then we will have a whole pool of various epitopes, which is detected by resulting antibody. If a peptide, which is 7 to 30 amino acids, is injected, automatically the amount of epitopes recognized by such an antibody is going to decrease. Agrisera produced antibodies which are reactive in thousands of species. So some of our photosynthetic antibodies will recognize D monocots and cyanobacteria diatom mosses. And many of antibodies to very conserved proteins will have this capacity. However, if you are purchasing an antibody to species which is not listed on product info sheet, please remember that even if protein is of high conservation, it is very good to first ask a supplier and send them a sequence of a protein which you aim to detect for confirmation if a peptide used to elicit this antibody is actually conserved in the sequence from the species you are aiming to work with. This is extremely important, and such predictions are very accurate according to our experience. So you can save your very precious time in the lab by doing a sequence conservation check. And especially antibodies to photosynthetic proteins have this possibility that they will react with a very broad species range. And we have developed around hundreds of such antibodies, which are called global antibodies and allow multi-species detection and quantification. So in case of, for example, Rubisco antibody, which is one of the most published Rubisco antibodies in the world, a very short peptide has been chosen, which is conserved in a wide species range. And this antibody, combined with a protein standard, will allow for quantification of Rubisco using a quantitative Western blot technique. This blot shows quantification of another photosynthetic protein of PSBA. These are samples from different cyanobacteria, and here are samples of a standard, which will allow to create a standard curve for quantification. So following protein standards are available mainly for photosynthetic proteins, and they are also part of the antibody sets, which contain both antibodies and respective protein standards, and will allow quantification. These sets are called photosynthetic, educational, and Rubisco quantification. Let's go back to antibody production. When it comes to an animal species which is chosen for antibody production, we have different possibilities. And here I have ordered them in respect of amount of serum which can be obtained from a given species. Please remember that polyclonal antibodies will recognize a pool of several different epitopes, while monoclonal antibodies, which nowadays can be produced in not only mice but also rabbits, rats, and chickens, will recognize only single specific epitope. This is a big difference which has an importance in terms of versatility of such an antibody and their stability. If monoclonal antibody loses activity, then such preparation is simply not active anymore. When polyclonal antibody is deteriorating, it is a process. It is still going to be other antibodies binding to other epitopes, which will give a reaction. When it comes to antibodies produced in birds, they are very good systems to obtain large amounts, as antibodies are in chickens localized in egg yolk, and they are provided as purified from egg yolk. So for a standard research lab, usually chicken or rabbit produces enough of antibody to fulfill amount of antibody needed for conducting plant experiments, while goat, donkey, or llama are used by diagnostic companies or for production of secondary antibodies where large amounts of antibody are required. Here you can see standard immunization programs for different species, which we conduct at Agrisera. And please remember that in a serum preparation which you're receiving, if antibody is not purified, it is only 1 to 10% of total immunoglobulin fraction, which will be against specific target which has been injected. So it's only 1 to 10%. Why should we inject not one but 2 animals? The reason for it is that each of animals have very specific immunological response. In our antibody production facility, the animals are not getting sick. If it happens, it's a very rare case. This is another reason to inject not only one animal. As you can see here, these 2 rabbits were injected with the same target, and it resulted in very different immunological response. So this rabbit produced an antibody only against one isoform, while the second rabbit covered much wider range of isoforms of this antigen. Before anything is injected, so-called pre-immune screening should be conducted. The reason for it is that animals are eating plant-based diet, and very many of them will produce antibodies against Rubisco, the most abundant protein on Earth. Here on this blot, you will see samples of Arabidopsis thaliana number 1 and barley number 2, which were checked against pre-immune serum from different animals on our farm. And as you see here, animal 1 and 2 already in pre-immune serum has reactivity against Rubisco. Also, animal 2 produced a very pronounced signal against the protein above 20 kilodaltons. So these animals are not really useful for antibody production against proteins in a range between 60 and 20 kilodaltons. The best here in this screen is animal 4. As a rule of thumb, we are saying that following such a pre-immune screen, one should choose animals where there is no signal plus minus 20 kilodaltons of a target protein. Which protocol is used for pre-immune serum screening? There is a standard protocol, but you should remember that here the incubation of pre-immune serum should not be done overnight, and the wells should not be overloaded with protein, as this will most certainly result in quite pronounced background. Once antibody is produced, there is a quality control conducted by ELISA test. So the wells of ELISA plate are coated with a free peptide, without KLH, without a carrier, and the sample of pre-immune versus immune serum is tested. But please keep in mind that such an ELISA quality control is not a confirmation of future antibody performance. The way the antibody will behave in endogenous sample needs to be determined. This antibody shows a very good titration curve, and most probably is going to work in the format of serum, and that actually has happened. The ELISA test will show you the highest dilution in which antibody can be used, which produces a positive signal. Immune serum, ideally when it's tested, should not produce any signal as when it's pre-immune, before antibody production is started, and the resulting immune serum should produce a strong signal in a desired molecular weight range for our target protein in investigation. Here you see an interesting example. This is one of photosynthetic proteins. An antibody was tested on different species, plant species, and the mobility of protein can vary between the species. However, the results can look in different ways. Here you can see results from 4 different laboratories. The same antibody has been sent to these laboratories, and they used Arabidopsis thaliana for testing. As you see here, obtained results with protocols which have been applied are very different. From lack of signal to completely to the blots filled with bands, and one laboratory actually in their conditions was able to produce a good signal. So one can ask yourself a question: is antibody not specific, or are binding conditions between antibody and target protein not optimal? Every step of Western blot is very important for final data quality. And here I combined examples of Western blots to proteins of very different abundances. Photosynthetic proteins are mostly quite easy to work with, and the loads, protein or chlorophyll loads per well, do not need to be so high, while proteins like argonauts require quite high load per well. It can also happen, here is an example of so-called garden blot, where we have samples from different plant species, that antibody will work with different efficiency in different species. It has also happened for us that antibody developed to a peptide which was perfectly conserved in a broad species range, did not work on a certain species unless urea was supplied in extraction buffer. So first most important thing to secure is amount of target in analyzed sample. And there are different prediction sample programs where you can follow localization of a protein in specific organ, as well as you can follow the predicted amount of protein in a tissue. As a rule of thumb, we do not produce antibodies to proteins which are present in tissues in amounts lower than 1.24 parts per million, because in such conditions there are low chances that the antibody will actually detect the target protein which is present in a tissue in way too low amounts. I would like to encourage you to use the databases to check in which specific organ has to be analyzed for the best success with the antibody. Also extraction, not only localization of a protein and choosing the correct organ is important, but also extraction, which has to be conducted in a very prompt manner to prohibit protein degradation. We have tested when you process different samples which, for example, up to 50, 100 samples are needed to be processed, the bead beater is a very good solution, as it will be doing extraction in a way which is very even between the samples. We have been comparing bead beater and sonication, which we used before, and we found that bead beater as a pretty efficient means of protein extraction, which is rapid and where overheating can be avoided. Also, when there's multiple of samples, these two protein extraction methods are quite time consuming and can also contribute to protein degradation. We have seen, for example, the differences in protein patterns in this classical method between sample 1 and 15 if extraction was conducted over a longer period of time. So please remember that the samples need to be of the best quality, and they need to be most optimally, they need to be fresh. Usually storage of a protein sample is not recommended and may result in protein degradation. Also, usage of protease inhibitors is recommended, and we have also seen cases when inhibitor of proteasome has to be included to keep the protein from degradation. Optimal extraction buffer can result in very different Western blot results. Here you can see two examples where the protein, where extraction buffer has been adjusted between sample 1 and 2, and the target band in applied extraction buffer. In a second experiment, there was a detection of the target protein. Here the seed material has been analyzed, and in extraction buffer A, the target had completely wrong molecular weight, while it was well preserved with the extraction buffer B. Also, sample denaturation conditions are very important. We are not recommending heating in 95 or 100 degrees for 5 to 10 minutes. Many proteins may simply aggregate in such conditions. A classical example is H+ ATPase, which will aggregate and there will be no detection of a signal on a Western blot. So our denaturation conditions, which we recommend, are 70 degrees for 5 minutes. Of course, membrane proteins may also require even lower temperatures for denaturation. On this blot, you see an example where a sample which was denatured did not yield a signal for the antibody, while used in native conditions, the signal appeared. Different laboratories have different protocols. Some include urea as a standard procedure. However, in certain circumstances, it may happen that an antibody will work without urea in the sample. I would like to encourage you also to look into fractionation. As for many proteins, especially of low expression, fractionation will be a method to concentrate the target and allow detection. As you can see here, based only on this result, one would say that the antibody is not working correctly at all, while fractionation has shown that it allows for concentration of a target protein and the correct signal is finally detected. Please note also that the background pattern will also change depending upon the fraction which has been used. Optimization of Western blot is also connected with a whole procedure. So we prepare the sample, we run a gel, and now we want to transfer it to the membrane. Successful Western blot is results in identical copy of a gel which is transferred to the membrane. And it is very important to keep in mind that depending upon molecular weight of a protein, it is more beneficial to, for example, use membranes of another pore size for proteins above 20 kilodaltons. Also, concentrations of methanol and SDS are different depending upon molecular weight of the protein. For high molecular weight proteins, wet tank transfer may work better than to semi-dry transfer. Will work better for proteins of lower molecular weight. So please consider that one and the same protocol will not work for different proteins, that it has actually there is a need for adjustments. We have been doing quantitative Western blots and realized that a sandwich where we have two gels is not optimal if proteins are to be quantified, because as you see here, the from gel 1 proteins have been transferred, while in gel 2 in a sandwich, some of the proteins have been left behind. So when working with quantification, the sandwich has to contain only one gel, which is blotted, not two. There is also a limitation how much protein can be actually transferred to the membrane. And when you work with unknown antibody, which you aim to characterize, you have to be very careful and all parameters have to be checked. So stain your gel following Western blot to see if any protein is left behind. Also, check your membrane following Western blot to see the separation, how the Western blot is working. You can easily check with a quick Ponceau stain, which can be applied to PVDF and nitrocellulose and is rapid and reversible. In this case, I wouldn't recommend to continue with the Western blot procedure, as we can see that there is a problem with protein separation. When we move on to the next step of blocking unspecific sites of the membrane to allow antibody to bind and recognize just a target protein, we can see here that there is some room for adjustments. This antibody is made to a protein, and here we have two blots. On the first blot, the blocking was rather short, and the concentration of the blocker was rather low. And by adjustment, by a simple adjustment, by increasing the time of the blocking and increasing the amount of the blocker, more unspecific binding sites have been blocked. So this antibody here, based on this result, was considered to be really not useful, while by adjusting blocking, one could obtain considerable improvement. Here we have another case. The blocking has to be extended overnight. So different antibodies may require different blocking conditions. And in some cases, we can go up to 10% milk, non-fat milk, as a blocker. Generally, in case of plant proteins, we have learned that BSA is not such an effective blocker, and we prefer to use non-fat milk. And of course, in some cases, one has to be careful, as using too high blocker concentration can result in overblocking. Antibody has to be also titrated for the best signal-to-noise ratio. And here you have two examples, two different protein loads and dilution combined with different dilutions of the primary antibody. So once we obtain a correct target band, we can play around a little bit with protein load and antibody concentration to get optimal and clean Western blot. In some cases, some antibodies may actually produce higher background if they are incubated overnight. So in this case, you can see the same primary antibody, which was incubated with the membrane, in this case overnight, and it resulted in a high background signal. And in this case, only 1 hour room temperature incubation was required. So it is different for different targets. Also, exposure time. When you work with unknown antibody, it is good to extend the exposure time to simply check which bands will come up. Especially if you run in parallel a sample with recombinant protein and a sample from endogenous extract, the signal in the first sample will most probably come quite quickly, while a signal from endogenous protein can require longer exposure time. So when you're testing an antibody, I would recommend to extend the exposure time to see if the band is appearing after longer time. Then the parameters of protein load per well and primary antibody concentration, these parameters can be adjusted. Buffers and washes are also of importance. For most antibodies, we have seen that they perform well in TBST. However, some antibodies may require the other buffer. Also, for some antibodies, lack of blocker can lower the background, and it is very important to use clean containers and water of the highest quality. If you experience high background, you can test to check if changing to a clean container between each Western blot step will improve the situation. When we test an antibody which is produced at Agrisera, or if you order your custom production, it is very important to have the right system to validate antibody performance. In direct approach, there is a need for mutants to be included in a testing process. So ideally, a band in a deletion mutant is gone or should be gone. Sometimes we obtain such results where the band of a target protein appears in both wild type and a mutant. And what to do then? Such a mutant has to be checked. Sometimes the mutants are not properly prepared or reversed. Also, immunoprecipitation can be performed, but not all antibodies will be suitable for this technique. You need to remember that. And then a band can be analyzed on a mass spec. So these are the direct approaches if mutants are available. But in many situations, there may not be reliable mutants available. And in such situations, we apply an indirect approach, which means that we can neutralize an antibody with free peptide. There will be immunoprecipitation happening. The antibody will bind to the peptide, and a resulting supernatant, which is used on Western blot, will produce no band. We can also use negative controls, means samples from species which do not contain target protein. We can also conduct fractionation. And if we observe desired band of desired molecular weight in the analyzed fraction, that can also be an indirect confirmation that antibody is recognizing a right target protein. Here is a summary over indirect testing approaches. So a basic one, comparing pre-immune and immune serum, the most ideal situation. Of course, molecular weight of our target band may differ depending upon different modification which protein can undergo. Also, we can include recombinant proteins as controls. We can also use specific conditions which should increase our target protein. But please remember, these are all indirect approaches of antibody validation. And also, remember that Western blot is only a validation method for Western blot. Antibodies have to be validated separately for each technique. So if an antibody produces a clean pattern in Western blot, it does not mean that it will perform well in immunohistochemistry, because it all depends upon really antigen exposure, which may be different depending upon the technique. How to save time? There is actually such a possibility in case of Western blot, that there are certain tricks which can be done to actually shorten the whole procedure. For antibodies of high titer, a simultaneous incubation can be performed, so everything happens at once. Blocking, primary and secondary incubation happens at the same time. So we can achieve a good signal strength on the shortening considerably Western blot procedure by several steps, cutting down several steps. We offer an incublocker solution for that. And this is suitable for antibodies of high and moderate titer. Another way to save time is to use a simultaneous detection with several antibodies at the same time. Antibodies need to have comparable titer to be applied that way. Here you can see example of 3 antibodies combined together. Also, for many antibodies to proteins of high or moderate expression, we can use directly conjugated antibodies to horseradish peroxidase or alkaline phosphatase, and this will shorten the Western blot time. Also, it is of importance what secondary antibodies we are using, because there is actually difference between the secondary antibodies in their strength. So if antibody is used in dilution of 1 to 2,000 or 1 to 20,000, it is a considerable difference in how many tests it can be applied. Also, secondary antibodies used in higher dilution will less likely contribute to any background. We can use secondary antibodies with for different detection techniques, which can be chemiluminescent, chromogenic, or fluorescent. In our hands, chemiluminescent detection method for Western blot appear to be most sensitive. There will be difference between secondary antibodies also in their background level. Agrisera secondary antibody, goat anti-rabbit horseradish peroxidase conjugated, can be used in dilutions about 1 to 25,000, and it has been used on a very wide range of different plant and algal moss diatoms samples. There will be difference between detection reagents which are applied for Western blot. Here you have a comparison of 2 different chemiluminescent detection reagents which we offer. It is ECL Super Bright, which has extreme low femtogram detection range, and here we can see detection done with ECL Bright, which has a mid picogram range. These reagents are combined in a kit, which you can use depending upon expression level of your protein. So our ECL kit contains 2 different ECL reagents. We can apply different detection methods, and some laboratories will have parallel systems for detection. Here you can see an example of the same primary antibody on the same set of samples, which produce quite different results depending upon detection method which has been applied. In the left panel, we have chemiluminescent detection, and in the right panel, we have fluorescent detection. Of course, please keep in mind that these are different secondary antibodies used in this case, which can also contribute to the final result. If you have not working antibody, it is important to try to troubleshoot the protocol to find where it can be improved. Here you can see an example of Rubisco antibody, one of the results we have received from our customers before optimization and after optimization. Not everything is lost. If you work with custom antibodies produced to your target protein, there are different things which can be done, which I have talked about. If you work with catalog antibody, you should carefully check product information sheet, because this should contain different details on how an antibody has to be used. For some antibodies, type of a membrane will have an importance. As you saw, load per well can have an importance. Blocking, type of blocker. Also, if an antibody is to a protein of low expression, it most preferably should be affinity purified. Also, I would suggest if you work with unknown antibodies, which has not been characterized, then one should kind of overload the system. So use quite a lot of protein load per well, primary antibody in larger amounts, and overnight incubations. If a target band is detected, one can still optimize these conditions and fine-tune them. If antibody is still not working, you are welcome to send your Western blot image with all details to Agrisera Support, and we will help you to troubleshoot and find the conditions of the binding. All these details have to be provided. As you have seen in these presentations, each of them actually has an importance. I would like to mention a few words about good laboratory practice, which is very important when you work with antibodies. For short-term storage, we use minus 20 degrees, and long-term storage means several years, minus 80 degrees. However, please keep in mind that some antibodies are not suitable to be stored at minus 80 degrees, and therefore, if you purchase a catalog antibody, you should carefully read the instructions. Chicken antibodies we are storing for over 20 years in just 4 degrees with sodium azide, which is an inhibitor of bacterial growth. Also, some antibodies may not be suitable for delivery in dry ice because they can be inactivated by transport in dry ice. Reusing of primary or secondary antibodies is not recommended, especially if you are working with quantification. Also, if you ever decide to conduct or order affinity purification, please do not purify all material at once. It has happened that some antibodies may lose activity following affinity purification. Therefore, only a small part of material should be used in a first attempt. And also, please remember that there is no standard protocol which will work with all antibodies. Antibodies are a little bit like people. They are all different. So they may require different binding conditions depending upon the target protein which they are recognizing. I would like to also remind you that antibodies used and published should be published in a way which will allow other researchers to recognize which antibody has been actually used. This is especially important when there are quite many antibodies to the same target protein. As for example, Agrisera has 10 antibodies to Rubisco. If we have antibodies to tag proteins, many companies provide multiple of antibodies to tags. Even secondary antibodies should be cited with product number, especially if they are used in immunolocalization. It is very important to know which exactly secondary antibody was used If a result is without a background, please consider citing antibodies correctly. I would like to also show you a little bit technical resources which are available on our website, which you can use. We have different video tutorials which are covering protein extraction, Western blot, and also quantitative Western blot. There is also a possibility to obtain a poster for your lab which covers all 3 parts of antibody production process. You can also request a hard copy of Agrisera Western blot guide. There were some hard copies of that guide also available at this conference. If you have not been able to get it and you are interested, please request it on our website and we will post it to you. How to contact us? You are always welcome to contact us with questions regarding antibodies. You can do it directly on our website through our chat system, or you can send us an email. And if you have any questions regarding this presentation, please email them to me and I will be happy to reply. If you would like to receive a PDF of this presentation, please contact me by email. And at the end, I would like to thank you very much for your attention. I couldn't arrive for this conference in person, therefore this recording, which I hope you will find useful. And at the end, I would like to encourage you to visit the Global Plant Event Calendar, which we hope is going to be helpful to organize your conference attendance. There is over 140 meetings registered in the calendar this year, and there are many meetings registered already for next 2 years. So please feel welcome to use it. Thank you very much for your attention and good luck in your research. You're always welcome to contact Agrisera. Goodbye.